Is prokaryotic cell and eukaryotic cell. What is prokaryotic cell and what is eukaryotic cell? First of all, prokaryotic cell. And second one, eukaryotic cell. So, what is the difference between prokaryotic cell and eukaryotic cell? Prokaryotic cell. Actually, before starting about the prokaryotic cell and eukaryotic cell, I will give a small little bit explanation about this cell. Just observe here. So, it is the cell. It is the cell. Each and every cell contain the DNA. Each and every cell contain the DNA. DNA means deoxyribonucleic acid, double stranded polynucleotide chain. Double stranded polynucleotide chain called as a DNA. So, each and every cell, you can take plant cell and animal cell and bacterial cell and fungi cell. Which kind of cell? Okay, you can take any kind of cell. Each and every cell contain the DNA. DNA means DNA contains a very important role in the cell. Suppose for example, all functions of the cell depend on the DNA. It means DNA will use to order for some functions. Suppose for example, is there any proteins required? This DNA will use order to produce the production of the proteins for the ribosome, and this DNA will use another. Okay, in any kind of cells, DNA have a main role. So, what is the importance of DNA? What is the importance of DNA? Means each and every function of the cell, it is controlled by DNA. It means DNA is equal to principal. In our college or school, okay, principal, what is the role of principal? Principal will organize the entire college or entire school and entire staff. Yes or no? So, that is why here also DNA equal to our principal, sir. So, this DNA will organize it means will command each and every mechanism and each and every function in the cell. So, that is why each and every cell contains the DNA. Now, next question is, what is the location of DNA? What is the location of DNA? Location of DNA, nucleus. Location of the DNA, nucleus. Suppose for example, what is the location of the principals are? What is the location of the principals are? Means students will say, sir, location of the principals are headmasters are in our school or college, office rooms are. So, principals are have a separate chamber, separate room, yes or no? So, that is why here also inside of the cell, in plant cell and in animal cell, whatever may be, in the cell, nucleus is a location of the DNA. Nucleus is a location of the DNA. Nucleus is a chamber of the DNA. It means plant cell contain the DNA and animal cell contain the DNA and bacterial cell contain the DNA and all kind of cells contain the DNA. But observe here, in plant cell and animal cell, separate chamber is there. What is the separate chamber? Nucleus. In animal cell, separate chamber is there for DNA, nucleus. In plant cell also, there is a separate chamber for a DNA. What is the separate chamber name? Nucleus. This nucleus surrounded by two membranes. Here also nucleus surrounded by two membranes. But just observe here, it is a bacterial cell. Just once again, observe here, it is the cell. Inside of the cell, it is the cytoplasm. Inside of the cell, it is the cytoplasm. Exactly at the middle of the cytoplasm, animal cell. At the middle of the cytoplasm, nucleus is present. This nucleus surrounded by two membranes, two layers. Inside of the nucleus, DNA is present. So, here also DNA is present. Just observe here, animal cell. In animal cell, in plant cell. In animal cell, in plant cell, there is a separate chamber for the DNA. What is the chamber name? Nucleus. So, but here in this bacteria cell, there is no nucleus. In this bacteria, there is no nucleus. But don't say there is no nucleus. It is a wrong statement. Actually, in this bacteria, there is no nuclear membrane. Nuclear membrane is absent. Nucleus membrane is absent. But what is the location of DNA? Actually, what is the location of DNA? Nucleus. But here nuclear membranes are absent. Nuclear membranes are completely absent. But in this bacterial cell, DNA located in the cytoplasm. DNA located in the cytoplasm. Which kind of place? Nucleoid. Okay, location of the DNA in the bacterial cell called nucleoid. What is nucleoid? Nucleoid means location of the DNA in bacterial cell. Just observe here, cell A, cell B, cell C, plant cell, animal cell and bacterial cell. What is the difference between these three cells? First two cells have a separate nuclear membranes and they have a separate chamber for DNA. But in bacterial cell, there is no separate nuclear membranes. Nuclear membranes are absent. It means in bacterial cell, naked DNA is present. Naked DNA is present. It means DNA located in the cytoplasm. But in animal cell and plant cell, DNA separated with cytoplasm with the help of nuclear membranes. Here also there is no connection between the cytoplasm and nucleus. Why? Because here nuclear membranes are present. Here also nuclear membranes are present. But here nuclear membranes are completely absent. This DNA located in the cytoplasm. So these kind of cells called as prokaryotic cell. This kind of cell called as prokaryotic cell. Now, what is the definition of the prokaryotic cell? Prokaryotic cell means if a cell without nuclear membrane, that kind of cell called as a prokaryotic cell. Okay? What is the prokaryotic cell? Definition of the prokaryotic cell: if a cell without nuclear membrane or if a cell without nucleus membrane, these kind of cells called as a prokaryotic cell. So, example for the prokaryotic cell: bacteria, okay, bacteria, and some kind of algae, and also PPLO. PPLO means pleuropneumonia-like organisms. These are the examples for the prokaryotic cell. And examples for the eukaryotic cell and plant cell and animal cell. Plant cell and animal cell. Plant cell and animal cell, both of them belongs to eukaryotic cell. Both are they called as a eukaryotic cell. But here plant cell and animal cell also contain several differences. Once again I repeat the sentence. Animal cell belongs to eukaryotic cell, plant cell belongs to eukaryotic cell. Both cells are belongs to eukaryotic cell, but these two cells also have some differences. What are the differences between plant cell and animal cell? I will discuss in the next topic. But here, what is prokaryotic cell? Prokaryotic cell without nucleus membranes, the type of cell called as a prokaryotic cell. Simple definition of the prokaryotic cell: nuclear membranes are absent. Nuclear membranes are absent. In prokaryotic cell, nuclear membranes are absent. Okay, what are the another differences between prokaryotic cell and eukaryotic cell? Means in prokaryotic cell, nuclear membranes are absent. In eukaryotic cell, nuclear membranes are present. Okay, what about DNA? In prokaryotic cell, circular DNA is present. Circular DNA is present. But in eukaryotic cell, thread-like DNA is present. Thread-like structure is present. And also in prokaryotic cell, there is no stones. There is no stones in DNA. Okay, sir, what are the stones? Okay, I will explain about the stones in at the end of the unit. Unit ending లోపల మనం stones గురించి డిస్కస్ చేద్దాం. So, in prokaryotic cell, there is no stone proteins. Stone proteins are absent. It means nucleosomes are absent. Nucleosomes are absent. In prokaryotic cell, nucleosomes are absent. In eukaryotic cell, nucleosomes are present. Just observe here, nuclear membranes are absent, nuclear membranes are present. In eukaryotic cell, nuclear membranes are present. Here, circular DNA is present, thread-like DNA is present. Stones and nucleosomes are absent, stones and nucleosomes are present. Okay, number of chromosomes is 1. In prokaryotic cell, entire DNA converted into single chromosome. Okay, during the cell division time, this entire DNA converted into only single chromosome. But in eukaryotic cell, there are number of chromosomes are there. Suppose for example, in our human body 23 pairs of chromosomes are there, and each and every plant body contain the different kind of chromosome number. So here in eukaryotic cell chromosome number is more than 2, but in prokaryotic cell only single chromosome is present. It means entire DNA converted into single chromosome during the cell division time. And what are the another differences between prokaryotic cell and eukaryotic cell? Means in eukaryotic cell organelles are present. Cell organelles like mitochondria and also endoplasmic reticulum and Golgi apparatus and lysosomes and ribosomes and here also vacuole and chloroplast and mitochondria and also endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum and Golgi apparatus and lysosomes and ribosomes. So these are the cell organelles. In eukaryotic cell number of cell organelles are present, but in prokaryotic cell only ribosomes are present. So what is the unique feature of the prokaryotic cell and eukaryotic cell? Once again I repeat, what is the unique feature of the prokaryotic cell and eukaryotic cell? What is the unique feature of the prokaryotic cell and eukaryotic cell? Means in prokaryotic cell also ribosomes also present, in eukaryotic cell also ribosomes also present. In plant cell, in animal cell, in bacterial cell, in 3 types of cells. It means in both 2 types of cells, in prokaryotic cell, in eukaryotic cell ribosomes are present, but one small difference is there. What is the difference between prokaryotic cell ribosomes and eukaryotic cell ribosomes? Means in eukaryotic cell 80s type of ribosomes are present. In eukaryotic cell 80s type of ribosomes are present. In prokaryotic cell 70s type of ribosomes are present. 70s type of ribosomes are present. Sir, what is this number 80 and 70? Yes, I will explain about these measurements in the next class. So these are the differences between prokaryotic and eukaryotic cell. So now I am going to give a detailed explanation about the prokaryotic cell. Prokaryotic cell means okay, I will take the example of the bacteria. Prokaryotic cell. Suppose for example, if you can take one example of the prokaryotic cell, it's the bacteria cell. And before starting this topic, actually bacteria are number of types of bacteria are there. Suppose for example, some of the bacteria are spherical in shape. Some of the bacteria are spherical in shape. Some of the bacteria are rod shaped bacteria also is there. Rod shaped bacteria. This one also rod shaped bacteria. And also some of the bacteria are comma shaped bacteria also is there. And some of the bacteria are and chain like bacteria. It means spring type of bacteria also is there. And some of the bacteria they change their structure depend upon the environment. Just observe here, spherical shape of bacteria, coccus. If a bacteria contains the spherical shape, that kind of bacteria called as a coccus. What is the coccus? Coccus means round shaped bacteria, spherical shape of bacteria. And second one, bacillus means rod shaped bacteria. Rod shaped bacteria called as bacillus. Rod shaped bacteria called as a bacillus. And vibrio, comma shaped bacteria. Comma shaped bacteria called as a vibrio. Okay, comma shaped bacteria called as a vibrio. And fourth one, spirillum. Spirillum means more than one twist. What is the difference between vibrio and spirillum? Vibrio contain the less than one twist. Spirillum contain the more than one twist. More than one twist or spring type of bacteria. Spring type of bacteria called as a spirillum. And fifth type of bacteria, some kind of bacteria they change their structure depend upon the environment. So pleomorphic bacteria. Pleomorphic bacteria. Example for the pleomorphic bacteria, azotobacter bacteria, pleomorphic bacteria. Pleomorphic bacteria, they change their structure depend upon the environment. Okay, pleomorphic. Just observe here, coccus, spherical shape of bacteria. Coccus, streptococcus is a one kind of bacteria. Streptococcus means what is the structure of bacteria, shape of bacteria, spherical shape. Bacillus, do you know about the lactobacillus? Lactobacillus bacteria making a curd, production of curd. So bacillus, rod shaped bacteria, lactobacillus. What is the shape of lactobacillus? Rod shape. And Vibrio and Vibrio cholerae. What is the bacteria name? Vibrio cholerae. What is the shape of bacteria? Comma shape, less than one twist. And Spirillum means a spherical shape of bacteria. And pleomorphic bacteria will show the pleomorphism. Azotobacter bacteria. Pleomorphism means if a organism change their structure depend upon the environment, that kind of mechanism called as a pleomorphism. So pleomorphic bacteria, they change their structure depend upon the environmental situations. So these are the structures of bacteria. So different kind of structures are there. And here suppose for example coccus, majority of the bacteria, coccus does not contain any flagellum. Flagellum is locomotory organ. But bacillus contain the locomotory organ. What is the use of locomotory organ? With the help of this locomotory organ, with the help of flagellum, bacteria can move from the one place to another place. So one region to another region. And also here depend upon the flagellum also, bacteria are different kind of bacteria also is there. Some kind of bacteria contain the only single flagellum, some kind of bacteria contain the group of flagellum, some kind of bacteria contain the both sides of flagellum, some kind of bacteria contain the entire surface of the bacteria contain the flagellum. So depend upon the flagellum also, bacteria are different kind of bacteria also is there. So here especially rod shaped bacteria contain the flagellum, but majority of the coccus bacteria does not contain any flagellum. So how they can move from one place to another place? That depend upon the environment. If a bacteria present in the water or present in any medium, that bacteria moves from one place to another place with the help of water or wind, whatever it may be. So if you can take the structure of this bacteria, each and every bacteria contains the same structure. You can take coccus and bacillus and vibrio and spirillum and pleomorphic bacteria, whatever it may be, all kind of bacteria contain the same structure. So the structure of bacteria indicate the characters of the prokaryotic cell. So now here bacteria cell. So what is this bacteria? Bacillus bacteria. So each and every bacteria surrounded by a cell wall. Each and every bacteria surrounded by a cell wall. This cell wall made up of with peptidoglycans. This cell wall made up of with peptidoglycans. And after cell wall, the outside of this cell wall, one slime layer is present. Outside of the cell wall, slime layer is present. This slime layer made up of with glycocalyx. Slime layer made up of with glycocalyx. It is the slime layer. The slime layer present outside of the cell wall. So here how many layers are there? Two layers. One layer is a slime layer, another one is a cell wall. This slime layer made up of with glycocalyx and cell wall made up of with peptidoglycans. And after cell wall, inside of the bacterial cell, inside of the bacterial cell membrane also is present. This cell membrane made up of with phospholipids and associated with proteins. The cell membrane made up of with phospholipids and associated with proteins. Or what is the composition of the cell membrane? The cell membrane made up of with proteins and lipids. Just observe here, this entire bacteria covered by three layers. What are the three layers from inside to outside? Cell membrane and cell wall and slime layer. What are the layers surrounded by bacteria from outside the inside? Slime layer and cell wall and cell membrane. So these three layers cover the entire bacteria. Just observe here this point. This point means if you can take any point, the cell membrane will show some inner foldings. The cell membrane will show some inner foldings. This inner folding called as mesosome. this inner folding will called as mesosome. Sir, what is the function of the mesosome? This mesosome will participate in the binary fission. During the binary fission time, this mesosome will move towards the outside of the bacteria. This mesosome will automatically separate the plasma membrane from another plasma to another plasma membrane. This mesosome will separate the plasma membrane. After separation of the plasma membrane, the cell wall also will form between these two plasma membranes. One mother cell will converted into two daughter cells. So, what is the function of the mesosome? Mesosome will participate in the binary fission. It means mesosome helpful in the cell division of the bacteria. It is the mesosome. What is the structure name? Mesosome. And also, this bacteria contain the locomotory organs. These locomotory organs useful in the movement of the bacteria. So, bacteria moves from one place to another place with the help of locomotory organ. What is the locomotory organ? Flagellum. So, depend upon the flagellum also, depend upon the number of flagellum and location of the flagellum also, bacteria are different kind of bacteria also is there. So, these are the flagellum, locomotory organ. Now, here one small structure is there. So, this structure called as villi or fimbriae. What is the use of this villi or fimbriae? This villi or fimbriae useful in the attachment of the bacteria. With the help of this structure, this bacteria will attach to the surface. With the help of villi or fimbria, this bacteria will attach to the surface. So, this villi or fimbria useful in the attachment. And center place of the bacteria. So, center place of the bacteria, this entire place filled with the fluid-like structure. This fluid-like structure called as cytoplasm. But it is the bacteria. Bacteria means prokaryotic cell. Prokaryotic cell means there is no special chamber for the DNA. It means there is no special nuclear membranes. Nuclear membranes are absent. So, nuclear membranes are absent. That's why DNA located in the cytoplasm. Double stranded DNA located in the cytoplasm. So, it is the DNA, deoxyribonucleic acid. But this place, so this DNA located place called as nucleoid. Nuclear membranes are absent, but DNA located place called as nucleoid, and it is the DNA. But observe here, in some kind of bacteria, not in all type of bacteria, in some type of bacteria, small additional circular DNA is present. Actually, it is the main DNA. Major DNA, long size of DNA, circular DNA, double stranded DNA. But in some kind of bacteria, small additional circular DNA is present. The small circular additional DNA called as plasmid, not plastid. Plastid means chloroplast. That is a different topic. Here plasmid. What is the plasmid? This plasmid present in some kind of bacteria. This plasmid will useful in the bacteria. Suppose for example, ఈ plasmid ఎందుకు యూజ్ అవుతుందంటే with the help of plasmid, this bacteria will produce the antibiotics. With the help of plasmid, this bacteria will produce the antibiotics. And also, this plasmid have a large amount of importance in the biotechnology. Okay, in your second year botany, in 5th unit biotechnology, you can learn about this plasmid very well. Plasmid have a major role in the biotechnology, in the production of medicines, in the production of chemicals, whatever it may be, in the production of enzymes. So, this plasmid have a very, very important role in the genetic engineering and biotechnology. So, what is plasmid? Definition of the plasmid: in some kind of bacteria, small additional circular DNA called as a plasmid. In some kind of bacteria, small additional circular DNA is present. This type of DNA called as a plasmid. And also in this DNA, ribosomes also is present. Ribosomes also is present. Which kind of ribosomes are present? 70s type of ribosomes are present. In prokaryotic cell and eukaryotic cell, which one is the unique feature? Means presence of ribosomes. Presence of ribosomes in prokaryotic cell contain the ribosomes, eukaryotic cell contain the ribosomes. But prokaryotic cell contain the 70s type of ribosomes, eukaryotic cell contain the 80s type of ribosomes. So 70s type of ribosomes means prokaryotic ribosomes. But just look at here, these ribosomes during the protein synthesis time, these ribosomes are located on the mRNA, mRNA means messenger RNA. mRNA means messenger RNA. Here, what is the function of the ribosome? Function of the ribosome, protein synthesis. What is the function of the ribosome? Protein synthesis. Synthesis of protein is a function of ribosome. So during the protein synthesis time, these ribosomes will participate in the protein synthesis. That's why during the protein synthesis time, these all ribosomes are arranged on a mRNA. The sequence of ribosomes on mRNA called as polysomes. What are the polysomes? What are polysomes? Define polysomes. Polysomes means sequence of ribosomes on mRNA. Sequence of ribosomes on mRNA called as polysomes. These polysomes will participate in the protein synthesis. So this is the structure of bacterial cell. And in this bacterial cell and food material also is present, and some kind of small structures also is present. But these are the major parts and major explanation of the bacteria. Now I am going to give a labeling of this bacteria. So what is this? Flagellum. What is this? Flagellum. Flagellum is locomotory organ. So it is the slime layer. The slime layer made up of with glycocalyx or polysaccharides. With the cell wall. The cell wall made up of with peptidoglycans. Cell membrane means plasma membrane. And this one mesosome, plasmid and ribosome and polysomes and villi or fimbriae. So and DNA, deoxyribonucleic acid. Here mRNA also genetic material, but mRNA is a single stranded genetic material. RNA single stranded genetic material. mRNA messenger RNA. This mRNA contain the message. Which kind of message? Protein synthesis. Okay, protein related message. This mRNA contain the protein related message. That's why it is called as mRNA. Here DNA, deoxyribonucleic acid, double stranded DNA, and this place, location of DNA, it is called as nucleoid. Okay, what is the location of DNA in prokaryotic cell? Means nucleoid. So these are the features of prokaryotic cell. So the entire structure belongs to bacterial cell. So bacteria structure. Bacteria structures. So prokaryotic cell, features of the prokaryotic cell is completed. So in the next video we will discuss about the eukaryotic cell. In next video we will discuss about the eukaryotic cell. What is eukaryotic cell? And eukaryotic cell means plant cell and animal cell. So what are the differences between plant cell and animal cell? And what is plant cell? Okay, which kind of structure surrounded by plant cell? It means plant cell surrounded by cell wall. What is cell wall? What is cell membrane? What is nucleus? What is endoplasmic reticulum? What is rough endoplasmic reticulum? What is smooth endoplasmic reticulum? And what is Golgi apparatus? What are the lysosomes, ribosomes, and vacuole, and mitochondria, and chloroplast? So in the next video we will discuss about the each and every cell organelle in the plant cell. Okay, right, thank you very much.